Plant nutrition is the study of the chemical elements and compounds that are necessary for plant growth and also of their external supply and internal metabolism. In 1972, E. Epstein defined two criteria for an element to be essential for plant growth. The first criteria, in its absence, the plant is unable to complete a normal life cycle, or number 2, that the element is part of some essential plant constitute or metabolite. There is, in accordance with Liebig's law of the minimum, there are 14 essential plant nutrients. Carbon and oxygen are absorbed from the air, while other nutrients including water are obtained from the soil. Plants must obtain the following mineral nutrients from the growing media. The primary macronutrients, which is nitrogen, phosphorus and potassium. The three secondary macronutrients, calcium, sulfur and magnesium. The micronutrients or trace minerals, which is boron, chlorine, manganese, iron, zinc, copper, molybdenum, nickel. The macronutrients are consumed in large quantities and are present in plant tissue in quantities from 0.2% to 4% on a dry matter weight basis. Micronutrients are present in plant tissue in quantities measured in parts per million ranging from 5 to 200 parts per million or less than 0.02% dry weight. Most soil conditions across the world can provide plants with adequate nutrition and do not require fertilizer for a complete life cycle. However, humans can artificially modify soil through the addition of fertilizer to promote rigorous growth and increase yield. The plants are able to obtain their required nutrients from the fertilizer added to the soil. A colloidal carbonaceous residue known as humus can serve as a nutrient reservoir. Even with adequate water and sunshine, nutrient deficiency can limit growth. Nutrient uptake in the soil is achieved by cation exchange, where root hairs pump hydrogen ions into the soil through proton pumps. These hydrogen ions displace cations attached to the negatively charged soil particles so that the cations are available for uptake by the root. Plant nutrition is a difficult subject to understand completely, partly because of the variation between different plants and even between different species or individuals of a given clone. An element present at a lower level may cause deficiency symptoms, while the same element at a higher level may cause toxicity. Further, deficiency of one element may present as symptoms of toxicity from another element. The root, especially the root hair, is the most essential organ for the uptake of nutrients. The structure and architecture of the root can alter the rate of nutrient uptake. Nutrient ions are transported to the center of the root, the stele, in order for the nutrients to reach the conducting tissues, xylem and the phloem. The Casparian strip, a cell wall outside the stele but within the root, prevents passive flow of water and nutrients, helping to regulate the uptake of nutrients and water. Xylem moves water and inorganic molecules within the plant, and phloem accounts for organic molecule transportation. Water potential plays a key role in plant nutrient uptake. If the water potential is more negative within the plant than the surrounding soils, the nutrients will move from the region of higher solute concentration in the soil to the area of lower solute concentration in the plant. There are three fundamental ways plants uptake nutrients through the root. Number 1, simple diffusion occurs when a non-polar molecule such as oxygen and carbon dioxide follows a concentration gradient moving passively through the cell lipid bilayer membrane without the use of transport proteins. Number 2. Facilitated diffusion is the rapid movement of solutes or ions following a concentration gradient facilitated by transport proteins. Number 3. Active transport is the uptake by cells of ions or molecules against a concentration gradient. This requires an energy source, usually ATP, to provide molecular pumps that move the ions or molecules through the membrane. Nutrients are moved inside a plant to where they are most needed. For example, a plant will try to supply more nutrients to its younger leaves than to its older ones. When nutrients are mobile, symptoms of any deficiency become apparent first on the older leaves. However, not all nutrients are equally mobile. Nitrogen, phosphorus and potassium are mobile nutrients, while the others have varying degrees of mobility. When a less mobile nutrient is deficient, the younger leaves suffer because the nutrient does not move up to them but stays in the older leaves. This phenomenon is helpful in determining which nutrient in a plant may be lacking. Many plants engaged in symbiosis with microorganisms. Two important types of these relationships are the first one with bacteria such as rhizobia that carry out biological nitrogen fixation in which atmospheric nitrogen is converted into ammonium, and number 2 with mycorrhizal fungi which through their association with the plant roots help to create a larger effective root surface area. Both of these mutualistic relationships enhance nutrient uptake. Though nitrogen is plentiful in Earth's atmosphere, relatively few plants harbor nitrogen fixing bacteria, so most plants rely on nitrogen compounds present in the soil to support their growth. These can either be supplied by decaying matter, nitrogen fixing bacteria, animal waste, or through the agricultural application of purpose-made fertilizers. Hydroponics is a method for growing plants in a water nutrient solution without the use of nutrient-rich soil. It allows researchers and home gardeners to grow their plants in a controlled environment. The most important solution is the Hoagland solution developed by D.R. Hoagland in 1933. The solution consists of all essential nutrients in the correct proportions necessary for most plant growth. An aerator is used to prevent an anoxic event or hypoxia. Hypoxia can affect nutrient uptake of a plant because without oxygen present, respiration becomes inhibited within the root cells. The nutrient film technique is a variation of hydroponic technique. The roots are not fully submerged, which allows for adequate aeration of roots, while a film-thin layer of nutrient-rich water is pumped through the system to provide nutrients and water to the plant. Processes. Plants take up essential elements from the soil through their roots and from the air, mainly consisting of nitrogen and oxygen, through their leaves. Nutrient uptake in the soil is achieved by cation exchange, wherein root hairs pump hydrogen ions into the soil through proton pumps. These hydrogen ions displace cations attached to the negatively charged soil particles so that the cations are available for uptake by the root. In the leaves, stomata open to take in carbon dioxide and expel oxygen. The carbon dioxide molecules are used as carbon source in photosynthesis. Functions of nutrients. Although nitrogen is plentiful in the Earth's atmosphere, relatively few plants engage in nitrogen fixation, that is, conversion of atmospheric nitrogen into a biologically useful form. Most plants therefore require nitrogen compounds to be present in the soil in which they grow. Plant nutrient is a difficult subject to understand completely, partially because of the variation between different plants and even between different species or individuals of a given clone. Elements present at low levels may cause deficiency symptoms, and toxicity is possible at a level that are too high. Furthermore, deficiency of one element may present as symptoms of toxicity from another element and vice versa. Carbon and oxygen are absorbed from the air, while other nutrients are absorbed from the soil. Green plants obtain their carbohydrate supply from the carbon dioxide in the air by process of photosynthesis. Each of these nutrients is used in a different place for a different essential functions. Let us study about macronutrients which are derived from air and water. The first macronutrient, carbon. Carbon forms the backbone of many plant biomolecules, including starches and cellulose. Carbon is fixed through photosynthesis from the carbon dioxide in the air and is a part of carbohydrate that store energy in the plant. Hydrogen. Hydrogen also is necessary for building sugars and building the plant. It is obtained almost entirely from water. Hydrogen ions are imperative for a proton gradient to help drive the electron transport chain in photosynthesis and for respiration. Number 3 is oxygen. Oxygen by itself or in molecules of H2O or CO2 are necessary for plant cellular respiration. Cellular respiration is the process of generating energy-rich adenosine triphosphate, that is ATP, via the consumption of sugars made in photosynthesis. Plants produce oxygen gas during photosynthesis but produce glucose, but then require oxygen to undergo aerobic cellular respiration and break down this glucose and produce ATP. Now let us study the primary macronutrients. First is phosphorus. Phosphorus is important in plant bioenergetics. As a component of ATP, phosphorus is needed for the conversion of light energy to chemical energy during photosynthesis. Phosphorus can also be used to modify activity of various enzymes by phosphorylation and can be used for cell signaling. Since ATP can be used for biosynthesis of many plant biomolecules, phosphorus is important for plant growth and flower seed. Phosphate esters make up DNA, RNA and phospholipids. Most common in the form of polyprotic phosphoric acid in soil, but it is taken up most readily in the form of H2PO4. Phosphorus is limited in most soils because it is released very slowly from insoluble phosphates. Under the most environmental conditions, it is the limiting element because of its small concentration in soil and high demand by plants and microorganisms. Plants can increase phosphorus uptake by mutualism with mycorrhiza. A phosphorus deficiency in plants is characterized by an intense green coloration of leaves. If the plant is experiencing high phosphorus deficiency, the leaves may become denatured and show signs of necrosis. Occasionally, the leaves may appear purple from an accumulation of anthocyanin. But phosphorus is a mobile nutrient. Older leaves will show the first signs of deficiency. It is also useful to apply a high phosphorus content fertilizer such as bone meal to perennials to help with successful root formation. Let us now look at potassium. Potassium regulates the opening end and closing of stomata by potassium ion pump. Since stomata are important in water regulation, potassium reduces water loss from leaf and increases drought tolerance. Potassium deficiency may cause necrosis or intervenal chlorosis. Potassium is highly mobile and can aid in balancing the anion charges within the plant. Potassium helps in fruit coloration, shape and also increases its Brix. Hence, quality fruits are produced in potassium-rich soils. It also has high solubility in water and leaches out of rocky and sandy soils. This water solubility can result in potassium deficiency. Potassium serves as an activator of enzymes used in photosynthesis and respiration. Potassium is used to build cellulose and aids in photosynthesis by formation of chlorophyll precursor. Potassium deficiency may result in higher risk of pathogenesis, wilting, chlorosis, brown spotting and higher chances of damage from frost and heat. Let us now look at nitrogen. Nitrogen is an essential component of all proteins. Nitrogen deficiency most often results in stunted growth, slow growth and chlorosis. Nitrogen deficient plants will also exhibit a purple appearance on the stems, petioles and underside of the leaves from an accumulation of anthocyanin pigments. Most of the nitrogen taken up by plants is from the soil in the form of NO3, although in acid environments such as boreal forest where nitrification is less likely to occur, ammonium or NH4 is most likely to be the dominating source of nitrogen. Amino acids and proteins can only be built from NH4, so NH3 must be reduced. Under many agricultural settings, nitrogen is the limiting nutrient of high growth. Some plants require more nitrogen than others, such as corn, because nitrogen is mobile. The older leaves exhibit chlorosis and necrosis earlier than the younger leaves. Soluble forms of nitrogen are transported as amines and amides. Let us now study macronutrients which are secondary and tertiary. Sulfur. Sulfur is a structural component of some amino acids and vitamins and is essential in the manufacturing of chloroplasts. Sulfur is also found in the iron-sulfur complexes of the electron transport chains in photosynthesis. It is immobile and deficiency therefore affects younger tissues first. Symptoms of deficiency include yellowing of leaves and stunted growth. Calcium. Calcium regulates transport of other nutrients into the plant and is also involved in activation of certain plant enzymes. Calcium deficiency results in stunting. This nutrient is involved in photosynthesis and plant structure. Blossom end rot is also a result of inadequate calcium. Magnesium. Magnesium is an important part of chlorophyll, a critical plant pigment important in photosynthesis. It is important in the production of ATP, though its role is as an enzyme cofactor. Magnesium deficiency can result in intervenal chlorosis. Let us now study silicon. In plants, silicon strengthens cell walls, improving plant strength, health and productivity. Other benefits of silicon to plants include improved drought and frost resistance, decreased lodging potential and boosting the plant's natural pest and disease fighting systems. Silicon has also been shown to improve plant rigor and physiology by improving root mass and density and increasing above ground plant biomass and crop yields. Although not considered as an essential element of plant growth and development, except for specific plant species that is sugarcane and members of the horsetail family, silicon is considered a beneficial element in many countries throughout the world due to its many benefits to numerous plant species when under abiotic or biotic stresses. Silicon is currently under consideration by the Association of American Plant Food Control Officials for elevation to the status of a plant beneficial substance. Silicon is the second most abundant element in Earth's crust. Depending on the silicon content, they can be divided into three major groups, that is wetland, Gramineae wetland rice 10 to 15%, dry land, Gramineae sugarcane, most of the cereal spices and a few dicotyledon species which contain 1 to 3%. Most of the dicotyledons, especially legumes, contain less than 0.5%. The long distance transport of silicon in plants is confined to the xylem. Its distribution within the shoot organ is therefore determined by transpiration rate in the organs. The epidermal cell walls are impregnated with a film layer of silicon and effective barrier against water loss, circular transpiration rate in the organs. Silicon can stimulate growth and yield by several indirect actions. These include decreasing mutual shading by improving leaf erectness, decreasing susceptibility to lodging, preventing magnesium and ferrous toxicity. Let us now study the micronutrients. Some elements are directly involved in plant metabolism. However, this principle does not account for so-called beneficial elements whose presence, while not required, has clear positive effects on plant growth. Mineral elements that either stimulate growth but are not essential, or that are essential only for certain plant species or under given conditions, are usually defined as beneficial elements. Number 1 is iron. Iron is necessary for photosynthesis and is present as an enzyme cofactor in plants. Iron deficiency can result in intervenal chlorosis and necrosis. Iron is not the structural part of chlorophyll but very much essential for its synthesis. Copper deficiency can be responsible for promoting an iron deficiency. Molybdenum. Molybdenum is a cofactor to enzymes important in building amino acids. Involved in nitrogen metabolism, molybdenum is a part of the nitrate reductase enzyme. Boron. Boron is important for binding of pectins in the region of the primary cell wall. Secondary roles may be in sugar transport, cell division and synthesizing certain enzymes. Boron deficiency causes necrosis in young leaves and stunting. Copper. Copper is an important element for photosynthesis. Symptoms for copper deficiency include chlorosis. Involved in many enzyme processes necessary for proper photosynthesis. It is involved in manufacture of lignin, which is a component of cell walls. Involved in grain production. It is also hard to find in some conditions. Manganese. Manganese is necessary for photosynthesis, including the building of chloroplasts. Manganese deficiency may result in coloration abnormalities such as discolored spots on the foliage. Sodium. Sodium is involved in regeneration of phosphoenol pyruvate in plants. It can also substitute for potassium in some circumstances. Essentially, substitution of potassium by sodium in plants can be classified into 4 groups. The first group, Group A. A high proportion of potassium can be replaced by sodium and stimulate the growth, which cannot be achieved by the application of potassium. Group B, which is the second. Specific growth responses to sodium are observed, but they are much less distinct. Number 3 is Group C. Only minor substitution is possible and sodium has no effect. And the last one is Group D, no substitution is occurred. Sodium functions in metabolism. First one, impair the conversion of pyruvate to phosphoenol pyruvate. Number 2, reduce the photosystem II activity and ultrastructural changes in the mesophyll chloroplast, replacing the potassium functions, that is internal osmoticum, stomatal functions, photosynthesis, counteraction in long distance transport, enzyme activation. Sodium also improves the crop quality. For example, it improves the taste of carrots by increasing sucrose. Let us look at zinc. Zinc is required in a large number of enzymes and plays an essential role in DNA transcription. A typical symptom of zinc deficiency is the stunted growth of leaves, commonly known as the little leaf, and is caused by the oxidative degradation of a growth hormone auxin. Nickel. In higher plants, nickel is absorbed by plants in the form of nickel ion. Nickel is essential for activation of urease, an enzyme involved with nitrogen metabolism that is required to process urea. Without nickel, toxic levels of urea accumulate, leading to formation of necrotic lesions. In lower plants, nickel activates several enzymes involved in a variety of processes and can substitute for zinc and iron as a cofactor in some enzymes. Chlorine. Chlorine as a compounded chloride is necessary for osmosis and ionic balance. It also plays a role in photosynthesis. Cobalt. Cobalt has proven to be beneficial to at least some plants, but is essential in others, such as legumes, where it is required for nitrogen fixation for the symbiotic relation it has with nitrogen fixing bacteria. Vanadium may be required by some plants but at very low concentrations. It may also be substituting for molybdenum. Selenium and sodium may also be beneficial. Sodium can replace potassium's regulation and stomatal opening and closing. Let us now look at aluminium. Tea has a high tolerance for aluminium toxicity and the growth is stimulated by aluminium application. The possible reason is the prevention of copper, manganese or phosphorus toxicity effects. There have been reports that aluminium may serve as a fungicide against certain types of rot root. These are the micronutrients